Good morning, guys of grade 9. Today, inshallah, we're gonna pass to section 2 of chapter 6. We're still talking about the DNA, but in that section, inshallah, we're gonna know how the DNA works. So we know the structure of the DNA and how different scientists already they contribute their works together just to build up the model of the DNA, starting from Charles Jeff, Rosalind Franklin, Watson, and Crick, and we know now how the DNA looks like and what is the building units of the DNA. Today, inshallah, we need to know how this structure or how that DNA could work to show the different characteristics of a living organism or how to control all the different biological processes that happen in each cell inside your body. So let's start the idea here. As we said yesterday in the classroom, or even once we talked the last time in our session, we said that all living organs, they contain DNA, starting from bacteria up to the human. We have DNA, and that DNA somehow made of the same units which are built of nucleotides. So that's why we said that DNA could units all living organs, because all living organs, they have the same structure of the DNA, or they have DNA with the same structure, but it's different in the arrangement of nucleotides, the amount and the arrangement. To clarify the idea for you, just look here in that we have this somehow the ordered letters, and we think what are the kind of words that could you form from these letters. Okay, if I ask you, could you make just 3 words of each group of these letters or something like this, so you can find what could I predict. So the first one, it's not a must to use all the letters. You can use 3, 4, whatever of them. So in the first one, T, P, S, O, N, E, I, R, how we could use this, so we can make many like stop, spot, sun, okay, so on. You can find a bin, okay. So you have a lot of words that you could predict from this group of words, group of letters, sorry. And second one, you can get genes, for example, okay. So the idea here depends on in just few letters, okay, that found in different arrangement, okay, and in different orders, they could form many words, a lot of words. So the same idea about the 4 nucleotides of the DNA, A, C, T, and G. Once they are ordered, okay, in different ways, or that they are found in different repetitions and amounts in different orders, so we are going to have different instructions or different codes. Each instruction or codes, okay, they refer to certain trait or certain characteristics in the living organism. So here, let's get, by the end of this lesson, we should be able to explain the relation between the DNA, genes, and proteins. And we should know what the basic type, the basic types in making a protein. Finally, describe 3 types of mutation and provide an example of each gene mutation. And finally, describe 2 examples of uses of genetic knowledge. Let's start here. Are revealing, okay, DNA. We said that DNA is already, or all the cells, they contain DNA, whatever it's a unicellular living organism or prokaryotic living organism like bacteria, or a multicellular living organism or eukaryotic cellular, okay, or eukaryotic cell, that means the cell which contain nucleus like plants or animal cell. Both contain DNA, but there is a little difference here. So we're gonna find that always DNA is bonded, okay, around protein. It's coiled around protein, okay, to form kind of strands, okay, and then bundle more and more, okay, to make something called the DNA or chromosomes. But here we go, need to differentiate between 2 different types of cells, how the DNA look like inside them. So we said that we have something called prokaryotic cells. Prokaryotic cells, that there are cells without nucleus, as we explained last year. Okay, so prokaryotes or prokaryotic cells, they are cells which have no nucleus. So in that case, DNA will be free in cytoplasm. So if we just look here, example like bacteria, that's the only example, okay, for the prokaryotic cell. So we're gonna find we have just a circular DNA. If we just get magnified this DNA, we're gonna find the regular shape of the DNA, which we know the double helix, okay? So in that case, we just have 1 circular chromosome or 1 circular DNA that's already found free in cytoplasm, okay? That's in case of prokaryotes. But once we talk about the eukaryotic cells, okay? So eukaryotic cells or eukaryotes, okay, that means the cells which have nucleus, okay, or normal nucleus as we know, like, okay, plants and animal, or plant cell and animal cell. In that case, we're gonna find the DNA is just trapped inside the nucleus, okay. We have a certain organelle that pull. In that case, we're gonna find the DNA actually, okay, here is coiled around each other. They form this network or that tangled chromosomes over each other, they look like a spaghetti dish. If we just try to, okay, uncoil these chromosomes, we're gonna find a chromosome is a thread-like structure like that, okay? So that's here, each chromosome. So what the chromosome is made up of, if we get apart and we start to maximize it, we're gonna find it made of DNA, and actually this DNA is just, okay, coiled around a protein. So the thread part inside that is a protein in which DNA, the black, this one, okay, is coiled around, okay? So here we're gonna find that mean always DNA, because DNA is too long, okay, strands. These two long strands, they're supposed to be coiled around protein to be able to fit in shape of chromosomes that able to fit inside a small or tiny space like the nucleus. Okay, so that's how we, or how the DNA looks like inside the cell. So here, back to the presentation, we're gonna show the idea more here, guys. So we said like this, a gene is just a part of the DNA which is responsible for a specific trait. So let's take an example to clarify the idea here. Here, for example, that's one cell we picked from a skin cell. So it's an animal cell, eukaryotic cell. So as you can see here, we said that the diameter of normal skin cell is about 0.0025 centimeters. Okay, that means too small, that the diameter of the entire cell. The DNA must be just trapped here inside. Okay, the DNA, all your DNA is just found in that small part in the nucleus. So if we just magnify it or just to bear up under microscope, we're gonna find it's look like a spaghetti dish, a lot of chromosomes that are, okay, twisted around each other. We call this huge network of tangled chromosome called chromatin. If we get one chromosome out of that chromatin, just we picked one out, okay, so we're gonna find it's the chromosome, just a group of protein in which the DNA is coiled around. So these orange circles represent the protein, and the DNA is represented by this yellow line. So the yellow, that DNA is twisted around protein to be able to fit like clusters, okay, here that make it smaller in size. Like when you get a thread, okay, in your home. So you're always, you find that the thread is coiled around a cylinder, okay, cartoon cylinder, which make it easier to be packed in small space. So once we get the DNA out and make it magnified more, we're gonna know it, okay, we're gonna see it, okay, the double helix model as we said. Each part, for example, that part here, that part of the DNA represents a gene. This gene is responsible for a specific trait, okay, and so on, guys, like that, okay. So here we said that actually we said that genes, okay, or the part of DNA that carry the code, that certain arrangement of the nucleotides in a certain way, okay, A, T, T, C, G, G, blah, 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 whatever the code is. This code suppose it refer to a certain trait or make the cell do a certain function with a certain way. So the idea here, okay, how the DNA works, how that code would make the cell work. Actually, if you have a coded message, you will not be able to read this message until you have the key or you get the, how to decode that code. So that same idea had also in the cell. As we have a code in the DNA, actually the DNA will not be functional until it translated into a molecule that the cell could understand, because the cell can't understand this code. So that means that DNA must be translated into protein. Protein considered as the chemical compound which the cell understands. Most biological processes that happen inside the cell and most traits is already expressed because of protein. So protein considered as a chemical triggers, okay, that starts most of the chemical processes and help in formation or determination of some traits. So that's mean here, okay, the cell understand everything in form of protein. So it means here the code of the DNA must be translated into protein. But unfortunately, that the DNA is trapped inside the nucleus and it's hard to get out of it, okay, because it's too big molecule, and even the pores or the holes which found in the nuclear membrane is not that big to allow the DNA to pass out. Beside that, once I need to get a code of one gene, okay, I can't cut DNA to just send one gene outside, and in this case also I am not able to to pass all the DNA outside. So here, guys, again, we said that the DNA is not able to come out from the nucleus to the cytoplasm where ribosomes, the site of protein production, are found. So in that case, DNA cannot pass to the ribosomes because, as we said, DNA is too big, it can't leave the nucleus, and at the same time, I can't cut the DNA because they are the genetic material. I don't get the risk to lose some of them. They must keep them safe inside the nucleus. and also ribosomes cannot pass inside the nucleus. So how could we make this kind of passing the instructions or the code to the ribosomes to be translated into protein? In that case, we need a kind of help. We need to send a message. So here that's become the role of the RNA. So that means the instructions will be sent in a form of a message or modified message. We call it the RNA. RNA actually is a ribonucleic acid. It's can somehow look like DNA with a little differences. That RNA is shorter, just made of one strand instead of two in the DNA, which make it smaller that could easily pass in and out of the cytoplasm. And it's a temporary copy of DNA. Just we copy the instructions which found in the DNA in form of RNA till it passed to the ribosomes to be translated and then will be broken down. Also here, the RNA is made of nucleotides like DNA because it's a nucleic acid, the same nucleotides of DNA, except thymine is replaced by another nucleotide, which is uracil. So that means in RNA, we have 4 nucleotides, A, U, C, G. U stands for uracil instead of thymine in the DNA. So guys, here that the main idea of our lesson, okay, we are gonna stop at this point today. And next time, inshallah, we're gonna know in details how the translation process happens or how the DNA could be translated into protein. Please study this part well from your textbook, and if you have any question, you can contact me anytime. Thank you. Goodbye.